The last Bio 111, Lab Introductions, Part 5, will cover topics from chapters 14, 15, and 16. We are going to talk about DNA, and this will have to do with the labs called DNA structure and replication and transcription and translation lab activities. Please refer to chapters 14, 15, and 16 for more information. Let's first talk about why DNA is important. I will go ahead and list 3 reasons why DNA is important. So, first and foremost, DNA is the molecule of inheritance. This is the molecule that you inherit from your reproductive mother and father. DNA contains segments called genes, which code for traits that you've inherited. More specific, DNA codes for various proteins and molecules. DNA has a special code in order to assemble or synthesize various proteins and molecules that your cells and tissues need. Let's list a second reason why DNA is important. DNA can be isolated from specimens and analyzed to serve various purposes. Some examples of DNA being analyzed would be to solve crimes or establish paternity. A few other reasons that we would want to analyze DNA would be to determine genetic diseases and establish its evolutionary relationships. So, analyzing the specific sequence of DNA can help us determine a lot of things. Okay, so let's go ahead and repeat that again. We can analyze DNA to solve crimes. We can analyze DNA to determine the paternity. We can analyze DNA to determine various genetic diseases. And we can also analyze DNA to establish evolutionary relationships. DNA is a very hardy molecule and can be isolated from fossilized specimens. This could help us determine evolutionary relationships. Let's talk about a third reason that DNA is important. So we can also manipulate DNA molecules in order to force the production of various medical treatments. Some examples of medical treatments would be insulin and clot dissolving medications. This brings us to our next topic of what we call biotechnology. Let's go ahead and list the definition of biotechnology. Biotechnology is the manipulation of DNA for the benefit of humans or the benefit of other various organisms. Let's give an example of the manipulation of DNA. Perhaps you've heard of diabetes. In the case of diabetes, individuals may need to supplement a shot of insulin in order to have enough insulin in their bodies. Where does all this insulin come from? An example of where the insulin may come from that humans use in order to treat diabetes would be from the manipulation of bacterial DNA. Bacteria often have special small rings of DNA called plasmids that can be fairly easily manipulated in order to integrate specific genes of interest into the bacterial DNA. When the bacterial DNA have genes of interest, then they will be able to produce substances like insulin. Please do not forget that a gene codes for a specific trait. And humans have a very specific gene which codes for insulin. So, in an instance where the individual does not make enough insulin, then they'll have a disease called diabetes. Some individuals with diabetes need to take a supplement of insulin in order to maintain homeostasis or maintain their health. This insulin can be produced from DNA, bacterial organisms that are grown in a lab setting. These bacteria will have special DNA called plasmids that humans can manipulate the DNA within the plasmids in order to force the bacteria to produce medicines like insulin to treat diabetes. This is what we call genetic engineering. Anytime that the DNA of an organism is modified, we're going to call that genetic engineering. There is a process called gel electrophoresis in which DNA is going to be broken up into separated fragments and produce a special pattern of DNA fragments called a DNA profile. Let's say, for example, we wanted to solve a crime or we wanted to establish paternity. Oftentimes, in order to solve a crime or establish paternity, we will take a sample of DNA and we will analyze that DNA in order to solve the crime or establish paternity. In order to analyze the DNA, we will need to visualize the DNA. The process of gel electrophoresis allows us to visualize DNA. What is formed after gel electrophoresis takes place is something called a DNA profile, which previously has been called a DNA fingerprint. Let's look at two examples of DNA profiles before. Again, these used to be called DNA fingerprints. What we're looking at are bands of DNA. We can also call these fragments of DNA. So here is one DNA profile, and here's another DNA profile. The first DNA profile is going to be DNA taken from a crime scene as well as 3 suspects. So the DNA is first isolated from the crime scene and processed in order to form fragments of DNA. And although it looks like little dash marks, these lines are representing fragments of DNA that have moved through a pad of gel. The gel is similar to jello that you may eat, but this is, of course, lab gel that we use for biotechnology. In this case, if we look at the first DNA profile, we see that we have a DNA sample from the crime scene, and we have a DNA sample from 3 individuals who we are going to call suspects. A process called gel electrophoresis will take all of those DNA fragments, and the fragments will move, or what we call migrate, across a slab of gel. This will produce a pattern of DNA fragments or DNA bands. This allows us to visualize DNA in order to help us solve crimes or establish paternity. So in this case, we are basically looking for the DNA bands from the crime scene, and there are a lot of DNA bands. Some of them are more faint, some of them are darker. We want to match that to the DNA from a suspect. So as you can see, the DNA bands from the first crime scene does not match the DNA bands from suspect 1. So we're going to rule suspect 1 out. But we can look at suspect 2 DNA bands and we see that the pattern of DNA is exactly the same as the crime scene. Therefore, our DNA supports that suspect number 2 would be would have been at the crime scene. Suspect 3, again, does not, the DNA bands from suspect 3 does not match the crime scene. So we have now solved the crime and we are choosing suspect number 2. When DNA is first entered into this slab of gel, DNA will then move. So let's say we entered the DNA here, or we submitted the DNA sample here. The DNA fragments that we've previously processed will move through the gel. We call this movement migration. Longer DNA bands will stay closer to where we first entered the DNA into the DNA sample well. Shorter fragments of DNA will migrate farther through our pad of gel. This will all take place during the process called gel electrophoresis. In this case, we had 4 samples of DNA. We had the crime scene DNA sample, suspect 1, suspect 2, and suspect 3. When we add DNA into different sample wells, okay, we generate something called a lane of DNA fragments. So in our very first DNA profile, where we are trying to solve the crime scene, we have 1, 2, 3, 4 lanes of DNA. Let's look at our second example of a DNA profile. How many lanes of DNA are seen in the second DNA profile? Again, 1, 2, 3, 4. In the second DNA profile, we are attempting to use the process called gel electrophoresis to produce a DNA profile in order to solve or establish a paternity. So we have mom's DNA, and we're going to go ahead and add that to lane number 1. And we have the child's DNA, and we also have 2 individual males, and we're trying to solve which male, Bob or Larry, will be the biological father according to the DNA. So remember that the child's DNA bands have to match up with either mom or dad. So this first band here matches up with mom. The second band matches up with Larry. The third band from the child matches with both Bob and Larry. So we're not going to be able to use that band to establish paternity. And then our fourth band looks like it matches up with Larry. So the child has much more DNA that is matching up with Larry. So therefore, we are going to say Larry is the father. The child's DNA bands and Larry's DNA bands have the most matches. Let's look at another example of a DNA fingerprint below. Up so we can see. Okay, so in our next example, we have 1, 2, 3, 4, 5, 6 lanes in our DNA profile. So here's our DNA profile. And in this specific example, we are trying to solve a crime. So this is crime scene. So it says lane A is the control. Oftentimes, when a gel electrophoresis process is done, we have a control entered into the first lane in order to make sure the process is working effectively. I'm sorry, lane A is the control. Lane B is the DNA that was isolated and processed from the crime scene. So lane B, we're going to call that the crime scene DNA. Lanes C, D, E, and F are going to represent suspects number 1, suspect 2, suspect 3, and suspect 4. Can you guys determine which suspect was at the scene of the crime based on the pattern of DNA bands that we see in our DNA profile? So you got that right. It looks like suspect number 3. The DNA bands in suspect number 3 do match the DNA bands from the crime scene. So we're going to go ahead and list suspect 3 as our perpetrator. Okay, let's move on to the next. So the next question, which is question number 3, and your lab introduction handout says, if directed by your instructor, go ahead and view the gel that was done from the lab called gel electrophoresis. Let's make sure to do this in class. Please leave this blank because you will have to actually draw the DNA fingerprint that you see from our lab class into the picture, into the diagram. So make sure you bring this handout to class so that you can draw your DNA fingerprint in this area. We are now going to move on to study the basic information about DNA. Right here. Let's talk about number 4. What does DNA stand for? Okay, DNA is going to stand for deoxyribonucleic acid. DNA. And you guys can see where DNA gets its abbreviation from. Please do not forget that DNA falls into the category of organic substances called nucleic acids. Back from chapter 3, we talked about 4 major classes of organic substances. They were called carbs, proteins, lipids, and nucleic acids. DNA is in the category of nucleic acids. These are all organic macromolecules that are made of individual subunits which are connected together in order to form large polymers. Let's go ahead and review the monomer subunits that the cell would need in order to make up a DNA polymer. It turns out if the cell is building a DNA polymer, it is going to need an individual monomer subunit called nucleotide. Actually, it may end up needing thousands of nucleotides because DNA is a very large polymer or very large molecule made of thousands of nucleotides connected together. Each nucleotide has its own name. So let's go ahead and write down the names of the nucleotides. Cytosine, we're going to abbreviate that with C. Guanine, we're going to abbreviate that with G. Thymine, we'll abbreviate that with T. And adenine, we'll abbreviate that nucleotide with the letter A. So therefore, the 4 nucleotides that make up all DNA polymer molecules are C, G, T, and A. For number 6, we are going to draw a very basic diagram of a cytosine nucleotide in the space below. And then we are going to go ahead and identify specific structures or parts of the cytosine nucleotide. So all nucleotides are made of a 5-sided sugar, which we're going to call a pentose sugar. The name of the pentose sugar in DNA is going to be called deoxyribose. And this is in part how DNA gets its name, deoxyribonucleic acid. That's the first part of the nucleotide. The second part of the nucleotide is something called a phosphate group. And the third part of the nucleotide is called a base. So in order to make one nucleotide, we will need a pentose sugar, a phosphate group, and a nitrogenous base. This is basically a structure or a chemical that has a lot of nitrogen in it. It turns out that all nucleotides have this basic structure. All nucleotides have a pentose sugar. All nucleotides have a phosphate group. The only thing that is different for the 4 different nucleotides is the nitrogenous base. In cytosine, the nitrogenous base is made of 1 ring. In some other nucleotides, the nitrogenous base could be 1 or 2 rings that are fused together. Let me give you another example of a nucleotide. I will make the drawing at the bottom of the page. This is another basic diagram of a nucleotide. All nucleotides contain a pentose sugar, all nucleotides contain a phosphate group, and the only thing that is different between the nucleotides is the nitrogenous base. Nucleotides that are made of 2 rings that are fused together in the nitrogenous base are considered larger nucleotides. Those nucleotides are specifically adenine and guanine, which we're going to go ahead and call purines. Purines is the term that describes the larger nucleotides. Pyrimidines is a term that describes the smaller nucleotides. An example of a nucleotide that is smaller would be the cytosine that we drew in the space above. Cytosine has only one ring making up its nitrogen base or nitrogenous base, which is the third part of a nucleotide. So examples of smaller nucleotides are going to be both cytosine and thymine. They're going to be made up of one single nitrogenous ring. There will be a little bit difference in the chemistry. And two examples of larger nucleotides are going to be adenine and guanine, and they are going to be made up of two rings which are fused together. Let's continue on to question number 7 and talk a little bit more about DNA polymers. So recall that the nucleotides that are needed in order to make up DNA molecules are going to be A, T, C, and G. DNA molecules are going to be made of a specific sequence of bases. Therefore, we say that all DNA molecules have a unique base sequence. Let me give an example of a base sequence. I have listed a very just a base sequence that I thought of in my head, but the concept still stands. This is an example of a base sequence within a DNA molecule. So you guys can see it's made of adenine, cytosine, thymine, guanine, adenine, thymine. All DNA molecules have a very specific sequence of nucleotides, which we call a specific base sequence. So I want you to think about the base sequence in your DNA molecule or your DNA molecules, and I want you to think about the base sequence in my DNA molecules, and then I want you to think about the base sequence in a bacterial cell's DNA molecules. Do you think the base sequence in your DNA is more similar to my DNA or more similar to a bacterial cell's DNA? Hopefully you chose that your base sequence is going to be more similar to my DNA. The more related or the more similar that organisms are, the more similar their base sequences are going to be. Now, this may be an example of just a tiny segment of one DNA molecule. Remember that humans have 46 DNA molecules. And each DNA molecule can be made of hundreds or thousands of nucleotides. So, of course, this is just representing a very small section of a DNA molecule and the base sequence within that section of DNA. Let's talk about the structure of DNA. DNA is actually considered a double-stranded molecule. Each strand is going to contain a specific base sequence of DNA nucleotides. The strands are going to be what we call complementary to each other due to something called base pairing. Research was done on various molecules of DNA that were isolated and analyzed from various species of organisms back in the 1920s and the 1930s, and including the 1940s. And a pattern was found within all DNA isolated from various organisms. The pattern found was that the proportion of adenine making up the DNA was roughly equal to the proportion of thymine in DNA molecules from various organisms. And the proportion of cytosine was roughly equal to guanine. This means that DNA taken from a starfish or DNA taken from an amoeba or a plant, DNA isolated from vastly diverse organisms were analyzed. And scientists found a pattern where no matter where the DNA came from, no matter which organism the DNA come from, the amount of A, which is called adenine in the DNA molecule, was approximately equal to the amount of thymine. Okay? And that same pattern was found for C and G. This suggests that adenine has some sort of relationship with thymine, and cytosine has some sort of relationship for guanine. So let me give an example of this. Let's say we isolated DNA from an organism and we wanted to analyze the composition of DNA from that organism. And we found out that 30% of the DNA molecule was made of adenine. If 30% of the DNA molecule was made of adenine, this means that 30% of the DNA molecule was also made of thymine, suggesting that there's some sort of relationship between adenine and thymine. This represents 60% of the DNA molecule. The other 40% would be 20% of the DNA molecule would be composed of cytosine, and 20% of the DNA molecule would be composed of guanine. So that represents 40%, and now we have accounted for the full 100% of the DNA molecule. In this case, what scientists have found was that there was something called complementary base pairing between A and T, and also complementary base pairing between C and G. Adenine was attracted to thymine within the DNA molecule. And anytime we have a force of attraction, a bond is formed. The type of bond that forms between our complementary base pairs is called hydrogen bonds. So let's say we have a strand of DNA and it has A, C. It turns out DNA is made of a complementary strand, and the bases on the complementary strand are going to form complementary base pairs compared to the other side. So therefore, on the complementary strand, we're going to find T, which pairs up with A. Again, T, and our last one is going to be G. So what's happening is that A and T are just slightly attracted to each other, and they form a bond that holds them together. That bond is called hydrogen bonds. It turns out that two hydrogen bonds form between A and T. So I'm going to go ahead and put two dots here representing the hydrogen bonds. So we can put two dots here as well. And three hydrogen bonds occur between C and G. So I'll put three dots between C and G. So please make sure you know that DNA is a double-stranded molecule. It's made of two strands, and the sequence on each strand is going to be complementary to each other. So we have 1, 2, 3 base pairs that we see in our DNA molecule diagram that I drew. The first base pair is A and T. The second base pair is A and T, and then the third base pair is C and G. So how many hydrogen bonds form between C and G? Again, that's going to be 3 bonds. How many hydrogen bonds form between A and T? Again, 2 bonds. One of the major goals in our lab activities today is that if I give you the base sequence of one strand of DNA, you should be able to tell me the complementary sequence on the other strand of DNA. So, for example, if the base sequence of DNA is AACGTC, then can you tell me the sequence on the other strand that would be complementary to that? So please make sure that you use the base pairs. So A pairs up with T. Okay, we got T here. The next one is T. Since we have A, C is going to pair up with G. G is going to form a base pair with C, A, and G. So that would be the complementary base sequence on the other strand of DNA. And you will want to make sure that you understand that. If on an exam or quiz I said the sequence of DNA was AAC, and I said, please determine the complementary sequence, then you would write TTG would be the complementary sequence. The next major activity that we want to discuss will be something called DNA replication. So DNA replication is a process that's going to be necessary for cell division. Remember, we start off with an original cell, and that cell basically doubles in size and duplicates everything inside of it, including DNA by DNA replication, and then the cell divides to form 2 identical cells. In this case, we're more or less talking about mitosis, but we can be also talking about the very early stages of meiosis. Please make note that special activities happen in meiosis in order to shuffle the DNA up, unlike mitosis, where after mitotic cell division occurs, we produce 2 clone-like cells. I would like to go ahead and make a very basic diagram to show you DNA replication. So I'm going to go ahead and start off with my DNA molecule. And remember that the DNA molecule is double-stranded, right? DNA is double-stranded. So here's a strand, and here's a strand. Oftentimes, the DNA molecule, the DNA strands twist together to form a shape very similar to a spiral staircase. Okay, and in this case, DNA would be called a DNA double helix. That is the common anatomical structure when we refer to DNA. However, when DNA is in the process of DNA replication, it is necessary for the 2 strands to separate and pull away from each other in order to allow for the replication process to occur. So let's go ahead and list some bases found on the DNA. Please pause the video and see if you can determine the complementary bases on the other strand. So the first one is going to be G. And remember, there are 3 hydrogen bonds between C and G. Can you go ahead and determine the rest of the bases that would be found on the complementary strand of this DNA molecule? So please go ahead and pause the video to test yourself to make sure that you are comfortable doing this. Please recall how many hydrogen bonds that we have between A and T and C and G. There are many enzymes involved in DNA replication. We are only going to list two. Please make sure to refer to the online textbook in order to determine various other enzymes that are involved in DNA replication. The first enzyme that we're going to talk about is DNA helicase. DNA helicase is essentially used to break all the hydrogen bonds between the two strands of the DNA molecule in order to separate the strands away from each other. This is sometimes referred to as unzipping the DNA molecule. So in this case, you see my G and C right here do not have a hydrogen bond. So it appears that DNA helicase has broken all the hydrogen bonds in order to separate the two strands here. DNA helicase will continue breaking all the hydrogen bonds for the entire DNA molecule. So remember, we're only looking at a segment of the DNA molecule, and the DNA molecule can extend for hundreds or thousands of nucleotide bases. DNA helicase will continue breaking the hydrogen bonds until the entire DNA molecule has been replicated. Our next activity, the next enzyme that we want to note will be an enzyme called DNA polymerase. DNA polymerase is an enzyme that is involved in catalyzing the reactions that basically build DNA molecules. Your cells have free nucleotides just floating around. Thymine, guanine, cytosine, adenine, just floating around individually. And when the DNA is undergoing DNA replication, new DNA polymers are going to be built. In order to build new DNA polymers, we need to take those individual nucleotides and connect them together to form new DNA polymers. I'm going to go ahead and use my purple highlighter so that I can highlight the original, or what we call the old or parental DNA strands. These basically serve as a template for the cell to build brand new DNA strands. What DNA polymerase does is it basically joins free nucleotides together in order to build strands of DNA which are brand new. I'm going to use the pink highlighter in order to show you a brand new DNA strand that is built based off of the original sequence from the parental strand. Oops. I did it again. Can you guys go ahead and build the new strand that would occur on this side if we are using this old strand as the template strand in order to build the new strand? Sorry, there's only 2 hydrogen bonds between A and C, and then there's 3 hydrogen bonds between G and C. So what I've highlighted in purple are considered the brand new DNA polymer strands that are built due to the presence of an enzyme called DNA polymerase. So just think about DNA polymerase. Basically, here are our new DNA polymer strands. So I want you to take a moment and look at the 2 molecules of DNA, this molecule compared to this molecule, to go ahead and determine if they are genetically identical. And by identical, I mean that they have the same exact base sequence. So if we look at the first one, GC, we are the same on this side, GC, GC, GC. Here's our third base pair, TA, TA, and we have our fourth base pair. They are exactly identical. This process will continue along the whole DNA molecule until the entire DNA molecule is replicated. This process is what we call semi-conservative, because the old DNA strands are maintained while the new DNA strands are being built. So basically, since we combine an old strand plus a new strand in order to duplicate the DNA or produce an identical DNA molecule, then we call the process semi-conservative. So again, just to review, okay, DNA kind of looks like a double-stranded molecule where there are bases. There's a specific base sequence, okay, on both strands, and those base sequences are complementary towards each other. Hydrogen bonds form between the 2 strands, and this keeps the 2 strands of the DNA molecule close together in order to maintain the structure of the DNA molecule, or the anatomy of the DNA molecule, which is a double helix. When the cell is about to undergo cell division, a process called cell replication or DNA replication has to occur. So during that DNA replication process, the strands, which are normally held together to form that spiral staircase shape, basically will be separated apart by DNA helicase. And then this enzyme, DNA polymerase, will be used in order to build brand new DNA strands, which are complementary to the old parental DNA strands. We need to move on to our next topic, which includes a similar molecule called RNA. So we are on question number 10. What does RNA stand for? RNA stands for ribonucleic acid. Ribonucleic acid. That's a little bit different than DNA, which is deoxyribonucleic acid. RNA is going to be important because it is needed for the process of protein synthesis. Proteins are very important to organisms. For example, humans have at least 23,000 different proteins. Some examples of proteins that you have probably heard about before would be hemoglobin, insulin, collagen, elastin, antibodies. These are all different types of proteins, and they all have their own specific and important function. RNA is an important molecule because it is absolutely critical and necessary for the process of building these important proteins. There is a name that describes the process of protein synthesis, and it is going to be called gene expression. The difference between DNA and RNA is that DNA contains the genes which code for the assembly of various proteins, but RNA is physically involved in assembling and synthesizing the proteins. RNA and DNA are similar, but they do have some differences. One of the first major differences is that RNA is a single-stranded molecule. And DNA is double-stranded. DNA is going to be a larger molecule because it is double-stranded. RNA will be a more slender type of molecule because it is single-stranded. RNA will contain a pentose sugar called ribose. That is a little bit different than DNA, which contains a pentose sugar called deoxyribose. Again, RNA is in that category of substance called nucleic acid. However, this is how it gets its name, RNA. However, it is similar but also different than DNA, which is also a nucleic acid. Recall that DNA is a polymer which is made of nucleotide subunits that are connected together. RNA is the same. In order to build a polymer of RNA, we need nucleotides, except we're going to call them RNA nucleotides. The nucleotides seen in RNA are very similar to DNA, and there is one difference. RNA will contain a base called uracil. So recall that DNA is made of A, T, C, and G nucleotides. RNA contains A also, but instead of thymine, RNA molecules have a base called uracil. RNA also contains G and C. So the molecules are similar, but there are some significant differences between RNA and DNA molecules. For question number 13, we are going to draw a very basic diagram of a cell, and we will show the nucleus within the cell and the DNA and various types of RNA molecules that are involved in gene expression. Our next major goal is to talk about this process called gene expression and involve the steps. To understand the steps involved in this process. So we're going to go ahead and use a eukaryotic cell in order to demonstrate the process of gene expression. So remember, the goal of gene expression is to build a protein. Let me give you an example. Let's say that you have consumed some food and now you have extra sugar floating around in your blood. Your body detects the extra sugar and it tells cells in your body to make insulin. Insulin is a type of protein. The gene that codes for insulin is on the DNA molecule. Now the cell wants to express the gene that codes for insulin because the body needs insulin. So the goal of the cell, or the job of the cell, is to build the insulin that the body needs. The very first step is to find the gene that codes for insulin on the DNA molecule. After we find the gene that codes for insulin on the DNA molecule, we want to express that gene. Expressing the gene means that we want to basically go from the code that is written within the gene to the actual physical protein that the body needs. Since we are talking about a eukaryotic cell, like a plant or animal cell, I'm going to go ahead and draw this big round structure, which is going to be called the nucleus. And please remember that in a eukaryotic cell, that DNA is trapped inside of the nucleus. I will use purple to represent the DNA molecule. Even though I say blue, red, and green, I'm going to go ahead and use various colors based on the highlighters that I have. So here's the DNA molecule. And the DNA molecule contains genes which code for the proteins that we want the cell to build. In chapter 4, we talked about certain parts of the cell, and this ooey gooey area that is in between the nucleus and the plasma membrane is going to be called the cytoplasm. The cytoplasm in a eukaryotic cell has a bunch of different structures, which we're going to call organelles, and other cellular structures. For example, there are going to be these dot-like structures called ribosomes found out in the cytoplasm. And back in chapter 4, our goal was to identify the ribosomes in the cell and then list the major function. The major function of the ribosome was that the ribosomes are the sites or the literal locations where the proteins are going to be synthesized. We have a little bit of a physical barrier problem for our eukaryotic cell because the DNA is a very large molecule. And remember, here's the gene. A gene is a segment of DNA. So this is the gene that is going to code for the protein that we want. Okay, so here's our entire DNA molecule, and segments of the DNA molecule are called genes. Let's say we want to express this gene. It codes for insulin, and the body needs some insulin. So what we're going to do is we're going to try to express that gene. Okay, if DNA could move out of the nucleus and give that code or that gene code to the ribosome, it would, but it cannot. DNA is a very large molecule and it's more or less trapped inside of the nucleus during this process. So now we have to build a molecule that we call a messenger. It's going to have to copy that gene code. And then since it's a very small molecule, it's going to squeeze out of the pores or the tiny openings of the nuclear membrane and present that code to the ribosome. So this molecule, which I'm going to highlight with the pink highlighter, is going to be called a messenger RNA molecule. The goal of the messenger RNA is to copy the gene code and then physically move out of the nucleus and present that code to the ribosome. So that is our messenger RNA molecule, which is sometimes abbreviated mRNA. Its job is to copy the code of the gene from DNA. And then since the molecule is a very small molecule, it can squeeze out of the nucleus and deliver that code to the ribosome. Since the ribosomes are responsible for building the proteins, okay, they're just waiting for a code from the messenger RNA molecule. Want to go ahead and note that the messenger RNA copies a complementary code of the gene, not an exact code of the gene. And we're going to go ahead and talk about that in a little while. We now have to talk a little bit more detail about what the ribosome does. At this point, we said the ribosome is the site for protein synthesis. Let's go ahead and add on a little bit more detail. In order to add on more detail, we have to talk about proteins. So remember that if the cell wants to build a protein, it's going to need that individual monomer subunit called amino acids. So when the ribosomes are synthesizing proteins, they are essentially connecting amino acids together in a specific order of amino acid based on the code from the original gene of DNA. I want to go ahead and note that ribosomes are joining the amino acids together. They do not have the amino acids stocked at the site of the ribosome. So we have to have another molecule enter our play of activities of what's going on. And our other molecule is going to be called transfer RNA. I'm going to use a yellow highlighter to highlight the transfer RNA molecules. Okay, so transfer RNA, which is abbreviated tRNA, is actually going to deliver the specific amino acid molecules. So the amino acids are ultimately this dot right here. That's going to be the amino acid. And then the rest of that molecule is going to be an RNA molecule called transfer RNA. So we have a bunch of different molecules that are involved when the cell builds a protein. This is a very particular order of events that occurs. The first major event is we need to find the original gene and make a complementary code of that. Okay, that first event is called transcription. Transcription is the process of building a messenger RNA molecule, and that's going to occur in the nucleus of a eukaryotic cell. The next major step and the last step is going to be called translation. And this is going to be when the final protein product is going to be built. These two steps are the two steps that are involved in gene expression. During gene expression, DNA, mRNA, tRNA, and the structure of the ribosomes are going to be all involved in working together in order to build the protein, which is the goal of the activity of gene expression. So let's go ahead and list on the bottom the types of RNA molecules, and it's just basically a summary of the RNA molecules that we have already talked about. The types of RNA molecules are going to be called messenger RNA. And you can see its function listed above in pink. The second type is called transfer RNA, abbreviated tRNA. And you can see its function listed above with the yellow highlighter. And our third RNA molecule that we want to just go ahead and mention is called rRNA, which stands for ribosomal RNA. This molecule is a structural component necessary for the ribosome. Just like the college is made of a framework of wood, maybe, the ribosome structures are also made of a framework, but the framework is going to be called rRNA. So basically, rRNA is simply a structural component of a ribosome. Let's go ahead and add a definition for gene expression. Okay, so the definition of gene expression, the process by which the information encoded in a gene is turned into a function. What is the function? Protein synthesis. We're going to stick with that topic. An example of gene expression would be blood sugar goes up, and this triggers the body to make insulin. So the first thing the body is going to do is find the gene that codes for insulin. It will make a copy of that gene by way of making a messenger RNA molecule. And then the messenger RNA molecule will deliver that code to the ribosome, which involves the function of the transfer RNA molecules. And then the amino acids within the protein will be joined together to build protein. Two major steps involved in gene expression are going to be number one, transcription. We mentioned this step when we drew the diagram of the cell. And the second major step is going to be called translation. So transcription happens in the nucleus of a eukaryotic organism. And translation happens in the cytoplasm. And this is going to be at the site of a ribosome. Each stage, transcription and translation, has a goal. The goal during transcription is to make the messenger RNA molecule. The goal during translation is to make the actual protein that the cell needs, which in this case would have been insulin. What we are going to do now is something called sequencing. During sequencing, we're going to start off with the original DNA molecule, and then we're going to talk about how the original gene encoded in the DNA molecule turns into a chain of amino acids, which we call a protein. So let's go ahead to number 17, where it says example of gene expression. Okay, I don't have these colors, but I am going to use my highlighters again during this process. So what I'm going to do is first start off with giving a small segment of DNA and looking at the sequence within that DNA. So let's say the DNA molecule had this sequence. I can tell this is DNA because if there is thymine, then that means that there is going to be DNA. Remember that RNA molecules do not contain T, and instead they contain U. So I'm going to go ahead and highlight this in purple. This is my DNA molecule because I've used DNA the purple color to represent the DNA molecule. Okay, this is the gene. And this is the specific sequence within the gene, AAC, CTG. Gene sequences contain a sequence of 3 DNA nucleotides called a triplet, which will ultimately end up coding for an amino acid. So I see that we have one triplet in my gene sequence, and we also have another triplet. The term triplet is going to refer to a DNA molecule. Remember that each triplet will end up coding for an amino acid. So if we go back to our steps of gene expression, for example, if we go back to our diagram, we see that we start off with the gene, and the next step involved in the process is to make a messenger RNA molecule. So let's go ahead and make a messenger RNA molecule for our next step. So let's say we want to express this gene. It's a very small gene, but we want the cell to express it. The next step in expressing this gene will be to make the messenger RNA molecule. And I believe I used hot pink for the messenger RNA molecule. So I want to show you that when messenger RNA is built in the nucleus of a eukaryotic cell, it's going to actually be built based on a complementary sequence. So now we will have to determine what is complementary to A. It turns out that normally T is going to be complementary to A, but we're not going to write a T here because RNA molecules do not contain T. They contain uracil, which is symbolized by the letter U. So let's go ahead and write the complementary sequence for the messenger RNA molecule, and we'll start off with U because A is going to be complementary to U. Again, RNA does not have thymine, which is T. Instead, they have the letter U. So can you determine the rest of the complementary sequence? The second one is also going to be U because A is complementary to U. Then we're going to have G, G, A, and C. That is the sequence of the messenger RNA molecule. Now, the messenger RNA molecule contains a sequence of 3 RNA nucleotides, which is going to be called a codon. And here's another codon. The process of making the messenger RNA molecule is called transcription, and this is happening in the nucleus of a eukaryotic organism. Okay, so now the messenger RNA molecule will move out of the nucleus and present the code to the ribosome. And at the site of the ribosome, the messenger RNA molecule will encounter a molecule called tRNA. Okay, so tRNA molecules are individual molecules, so they're not necessarily a strand that like DNA and messenger RNA. So I'm going to draw a tRNA molecule. It kind of looks like a pitchfork at one end and a dot at the other end. They're individual molecules that are just floating around the cell and waiting to be put to work. So not only do we have the messenger RNA molecule moving to the ribosome, but the transfer RNA molecules will also move to the ribosome. At one end of the transfer RNA molecule is a sequence of 3 nucleotides, which will be complementary to the messenger RNA. So this one is going to be A, C, because A is complementary to U, A is complementary to U, and C is complementary to G. We call this sequence of 3 nucleotides found on a tRNA molecule an anticodon. Can you figure out the anticodon for the second transfer RNA molecule? Going to be C, U, G, because C is complementary to G, U to A, and G to C. Each transfer RNA molecule contains an amino acid. So here's the amino acid at the other end. Our next major goal is to determine the name of each of these amino acids. In order to determine the name of the amino acids, okay, we're going to go ahead and we're going to actually use the messenger RNA codons. So it looks like the first messenger RNA codon is UUG. It turns out that scientists were doing research in this area and trying to determine how messenger RNA coded for specific amino acids. And scientists came up with a table of all of the amino acids that we need and all of the messenger RNA that code for them. So, for example, our first codon is UUG. So we're going to try to find that codon sequence in our genetic codon table. Okay, so this table is located in your lab manual. And we're again, in this example, we're looking for UUC, right? That's our example. I'm sorry, UUG. That was our first codon. Okay, I want to go ahead and mention that we studied it. Scientists in the past studied it in this way. And this is why we built a table that represents all of our amino acids, for example, leucine, isoleucine, methionine, valine. Okay, we even have tryptophan, which is abbreviated TRP. Okay, we have 20 different amino acids that our body needs to build all of the proteins, okay, that we need. Okay, and when scientists studied different proteins, they wanted to understand how did DNA code for the specific proteins that are found in, the specific amino acids that are found in proteins. And scientists ended up looking at the codons in order to determine the specific names of the amino acids. So when you are going to determine the name of the amino acids that will ultimately be in your protein, you want to go ahead and look at the messenger RNA codon. So our first messenger RNA codon is UUG. I'm going to go ahead and find that in the table of codons. So I see, here we go, UUG is right here. And it codes for amino acid called, well, it's abbreviated LEU, but that stands for leucine. So what you're looking at in this table are the abbreviations for the names of the amino acids. Okay, so our first one was UUG. I'm going to go back to my diagram and I'm going to add leucine as my first amino acid. Here's my abbreviation, but this amino acid that this transfer RNA molecule is carrying is called leucine. So that's the first amino acid in my protein. Can you guys determine the second amino acid in the protein? Again, please make sure you are using the codons in order to determine the amino acids. So you should be looking up GAC in the table of codons. I'm going to try to find GAC. So our next one is GAC. And that was from my previous page of notes. It looks like we have G's here and our A's are here. So GAC. Okay, here it is right here. That codon is gonna, here's our second codon. It looks like it codes for something called ASP. Okay, ASP actually stands for aspartic acid, which is just the name of another amino acid. You can very easily Google the codon chart to find the full names of the amino acids. Your lab book has this table, okay, within your lab manual, and these are the abbreviations for the name of the amino acids. So I'm going to go back to my original page with my notes, and I'm going to add aspartic acid as the second amino acid. Now I have determined both of the names of the amino acids in my protein. You guys, what's going to happen is that leucine and aspartic acid will be connected together by a bond at the site of the ribosome. So here's aspartic acid and here's leucine, and the bond that connects these amino acids together is going to be called a peptide bond. So at the site of the amino acid, peptide bonds are formed, okay, in order to connect amino acids to build a very large chain of amino acids, which we're going to call a protein. The function of the ribosome is to form peptide bonds to connect amino acids. This is why we say that the ribosome is the site of synthesis for proteins. I want to go ahead and mention the central dogma. Okay, the central dogma is the phenomenon, or basically a theory, or not a theory, I guess it's basically in science, we call it a central dogma, and it basically states that the flow of genetic information in any type of cell starts off with DNA and then goes to RNA. That could be your messenger RNA and your transfer RNA, and then ends up, okay, as a protein. This is what we call the flow of genetic information. The flow of genetic information, or the central dogma. In all cells that we know of, all cells go in this order in order to build proteins, and all cells need proteins. Plant cells, animal cells, our cells, bacterial cells, all of them need protein. In fact, even viruses, which are not alive, also need proteins. One of our targets in order to get rid of a virus is to stop the virus from making proteins. So I want you to realize that when you go on to microbiology, that you will definitely have to understand that we start with DNA, we go to messenger RNA, and then the cell goes to produce proteins. You'll have to understand the flow of genetic information. In addition to that, you should also be aware of the steps that are involved. This process called transcription builds messenger RNA, and translation basically happens at the site of the ribosome. Okay, these are going to be important things to understand when you move on to microbiology and you are in the chapter where you're trying to study how viruses infect and attack cells in a variety of ways. Okay, viruses are filled with some sort of genetic information. Okay, so sometimes that genetic information is DNA, and sometimes viruses contain RNA and not DNA. Okay, but basically, some viruses try to disrupt the flow of this genetic information in order to hijack or infect your cells. The type of virus that is going to go into your cell and manipulate the process of genetic flow and gene expression is called a retrovirus. Okay, a retrovirus tends to be a virus that is carrying messenger RNA, and the messenger RNA, as well as various proteins, move into your cell when the bacteria infects your cell and kind of messes up the flow of information in your cell. Ultimately, these retroviruses will end up using the messenger RNA to make viral DNA, which will be inserted into your DNA. So we don't really have to go into that one too much. Let's skip that one and let's move on to question number 19, which is mutation. Okay, mutation is going to be a change in the base sequence of DNA. So mutations are anything that results in a change in the base sequence of DNA. This can happen naturally, or this can be due to exposure to various substances. I want to assign you the activity of working on your own to investigate various types of mutations. So, some mutations may result in insertion of bases into the base sequence of DNA. Some mutations may result in some bases being deleted out of your DNA molecules, and so on and so forth. So, I want you to go ahead and please look up all these different types of mutations and be able to briefly describe them and distinguish them from each other. Thank you for watching the last biology introduction, part 5 for the semester.